In this video, we're going to learn about a special type of protein known as enzymes. Enzymes are biological catalysts, which means that they're functional proteins that speed up a reaction without being used up themselves. So just like a catalyst in chemistry, they help a reaction go faster because they're lowering the activation energy of the reaction, and then they themselves aren't one of the reactants, so they aren't involved in the reaction themselves aside from speeding up that rate. And enzymes are specific to their substrate, and the substrate is the substance that they act on. So in this image here, this green object is the substrate, the blue object is the enzyme, and then where they lock together is known as the active site. And when they form together and join together, they form the enzyme substrate complex. And what happens is once they've bonded, the enzyme acts on the substrate and turns it into products, and then it will release the products and they go off on their way, and the enzyme would be ready to act on another substrate. There are two commonly accepted models for enzymes. So one of them is known as the lock and key model. And in this model, we say that the enzyme's active site is the lock and the substrate is the key. And because the active site is the exact shape of the substrate it acts on, it only allows that substrate to bind. So only the correct key can fit in the lock. And this model explains an enzyme's specificity for its substrate. So again, you can see here the active site, the enzyme binding together, products being formed. So this is a breaking down reaction, so breaking something into two smaller components, and then the products are being released. Now the induced fit model suggests that the enzyme's active site will mold to take the shape of the substrate only after it started to bind. So it's kind of like us putting on a glove. So when you first put your hand into the glove, your fingers might not line up properly, but eventually you'll get it aligned once you have one finger fit in, and then the rest of the glove will go on quite easily. So that would be the idea of the induced fit model. The enzyme wraps around the substrate and then acts on it, and products are produced and then they're released. So there are many factors that affect enzyme function, and we will be conducting an investigation into this where you will select one of those factors and test it and see how it affects a specific enzyme. The enzyme we're going to be looking at is catalase. First of all, temperature will affect an enzyme action. So every enzyme has an optimal temperature for reaction, and at that point the reaction rate will be the fastest. Outside of those optimal ranges, you would have a slower reaction rate. And in fact, with enzymes, there is a point where at really high temperatures would actually be damaging to the enzyme, because enzymes are proteins and proteins can become denatured by high temperatures. So if we were to look at a graph of enzyme action, you would have, at low temperatures, a low reaction, and eventually it would increase as the temperature increases, but then it gets to the most optimum, and then it would start declining again. So a graph would look like an upside down bell or upside down parabola. pH also has an optimum range, so same thing. Outside of that optimum range, you would have lower reaction rates. So for instance, some enzymes that exist in our stomach, the optimum pH would be around 1 or 2, whereas most of the other enzymes elsewhere in our body would be around pH 7 or slightly higher than that. Also, another factor that affects enzyme function would be the concentration of the substrate or the enzyme, because the more substrate you have, the more chances that it's going to find an enzyme to bind to and get converted to products. Same thing, if you have more enzymes, it's more likely that it will find a substrate to act on and create products. So the higher the concentration of the substrate or the enzyme, the faster the reaction rate. Now, sometimes enzymes can be inhibited, and there's two different types of inhibitors. First of all, a competitive inhibitor is one that is similar in shape to the substrate, so it binds to the active site like this little orange blob right here, and then the actual substrate can't bind. So it's prevented from binding, so then the enzyme can't act on the substrate, and the substrate can't be converted to products. So this would inhibit or slow down the rate of reaction if there are inhibitors binding to the enzymes. The other type of inhibitor is a non-competitive inhibitor. So this is an inhibitor that doesn't bind to the active site, like here. Instead, it binds to a site called the allosteric site. So this is a site that's away from the active site. And then what it does is it changes the enzyme shape at the active site. And now the substrate can't bind because the shape has changed and it can't fit. So this is non-competitive inhibition.